Thank you very much for the kind introduction, and thanks to all of the organizers for bringing us together in this breathtaking, beautiful place. So I will talk today, and we'll start with a somewhat bleak and maybe provocative slide regarding the state of precision medicine. So if you look at this slide, you see that if you were unfortunate enough to be diagnosed with a new cancer in the US in about 2020, then you only had about a 13% chance of being eligible for a genome-targeted therapy, meaning that there's a large population of patients for whom we simply do not have these targeted inhibitors available that address their genomic lesions in their cancer cells. So there's a lot of unmet need. We need new drugs. We need new targets. Now, how do these, shining lights look like of targeted drugs that we hope to achieve? So you all are familiar with imatinib. This here shows a plot of survival in years, and on the x-axis, you see the year of diagnosis. And we have two cohorts of patients. So we basically have healthy controls that have the same age in blue, and we have CML patients, which you see here in yellow, which are 55-year-old female patients. And up to 1990, there was about a 25-year gap in remaining life expectancy for these patients compared to an age-matched cohort. But with the approval of imatinib, you see now that this gap in life expectancy has completely closed to zero by about 2010. So this is a drug that removes all of this gap in life expectancy, and that's what we should aspire to have in terms of targeted therapies. Now, if you look into the new approvals, so this is an average of all the 20 last years and all of the new approvals, you see that there's a median overall survival benefit of only 5 months for patients. So very different from this picture in the left where we gained 25 years of life expectancy. So this tells us that we should look elsewhere and maybe should look outside of the cancer cells themselves and look at the microenvironment. Drug development is getting harder over time. Here I use this famous example of the Moor's law, which shows how the transistor density on microprocessors has logarithmically increased over time. And in drug development, there is an inverted law, which is called Eroom's law, which you see here. And this tells you the amount of FDA-approved drugs per $1 billion in research and development spending. And you see here we have a logarithmic decrease, unfortunately. Whereas in the 1950s, there was a few dozen drugs approved per billion. Now it's less than 1. We had phases of regulatory tightening. This was the thalidomide scandal. And of course, we also had boosts. This was the age of biologics. But overall, this is a very sad picture. And also this tells us that we have to look elsewhere and look for new targets because the low-hanging fruits are mostly gone. 90% of all papers are studying 10% of genes in cancer, so it's very, very good that there's a lot of consortia such as dependency map that we've heard about that are basically using unsupervised screening methodologies to look at this large part of the dark genome that is usually not being studied. But of course, these screens, as we've heard from Brian today, are mostly studying cell autonomous effects, and we're going to miss cell interactions in the tumor microenvironment. So I don't have to convince this audience at all about the importance of the cellular diversity and the tumor microenvironment. But of course, if you consider the tumor in a traditional way as a bag of uniform cells, you have very, very limited cell interaction opportunities. But if you consider it as this ecosystem of different cell types, of immune cells, stromal cells, adipocytes, fibroblasts, then you have a lot of cell-cell interactions that could potentially drive the tumor in its growth. And of course, targeting these cellular interactions has huge clinical potential. You all are familiar with the interaction between a T cell and a tumor cell. If you block this PD-1, PD-L1 interaction, you can then reactivate the T cell to kill the tumor cell, and this has led to very meaningful, long-lasting clinical benefits in patients that had very dire perspectives before this therapy. And these cell-cell interactions were discovered by serendipity. And of course, we know that the PD-1, PD-L1 interaction and CTLA-4 were rewarded with the Nobel Prize a few years ago. And the picture has expanded. We know many more interactions nowadays, but we still lack a systematic way of testing cell-cell interactions in relevant contexts. And that's exactly what my lab is trying to work on. And of course, if we look at the picture of patient eligibility, you see here the picture looks better. So in 2019, almost 40% of all patients were already eligible to checkpoint inhibition. So we start with the hypothesis. Cell interactions can be involved in driving tumorigenesis. And with cell interactions, I mostly talk about a cell expressing a ligand in red that interacts with a receptor in yellow, so paracrine interactions and maybe autocrine interactions. And for the sake of this talk, I'm not going to focus so much on endocrine long-range interactions. So thanks to the revolution in single-cell genomics that we experienced in the last decade, we have very potent algorithms that can predict cell-cell interactions. Namely, we can just take these single-cell RNA-seq data sets, look at a given cluster. Is a ligand that we know from a database expressed, and is it cognate receptor expressed? And if so, these putatively interact. And if one of the components is missing, well, likely they're not going to interact. This is nice, and we have done this during my postdoc at the Itzkovich lab in the Weizmann, where we used human colon cancer that metastasizes to the liver. So we used these samples from patients and did single-cell RNA-seq, and we get these clusters, and then performed ligand receptor prediction. There's a whole bunch of different tools to do that. The problem is, if you do that not across just 2 cell types, but across all of the cell types in the tumor microenvironment, you get thousands and thousands of putative interactions. And this is not experimentally actionable, right? You don't know what to test. You have simply too many of those. This is just a computational prediction. But we don't know if these are causing a phenotype, or if they're just mere correlations that occur because of another phenotype. So how do we address this problem? There was a very elegant study by Martin Guillaume in Leuven that basically said, well, if a cell-cell interaction is important, it's likely going to be conserved across evolutionary time. So he not only studied a cell-cell interaction for a given phenotype in one context, but actually used different model organisms to see, out of, let's say, 10 candidates, which one is conserved across all of these models. This is very elegant, but unfortunately not really feasible for our topic of interest, which is cell interactions in colon cancer. So we argued with a different way of prioritizing. This is a huge simplification and can be easily criticized, but we say a lot of these paracrine signaling modules happen not in an infinite space, but rather constrained to about 200 micrometers. So it will be informative to know not only the nature of a given receptor ligand and where it's expressed, but also what's this 200 to 300 micrometer microenvironment that actually can be addressed by this molecule. So we focus on developing methodologies that can give us expression of single cells, of ligands and receptors, but also maintains the spatial relationship in this important informative diameter of 200 to 300 micrometers. So, 2 students, Jonas and Atti in the lab, are doing exactly this in the context of human colon cancer metastasis. So they took archival samples. Here you see the liver, the adjacent liver, and down here you see the metastasis. And you see this interface where these 2 tissue types meet. And we are now interested in ligands and receptors that might interact and cause some kind of tumor advancing phenotype, not anywhere, but only at this interface. So to get to those molecules, we used Visium spatial transcriptomics and basically performed 5,000 bulk RNA-seq experiments where we know the coordinates of each of the data points, and then reran this ligand receptor prediction specifically at the interface between the 2 types. Sorry for the resolution, which went a bit down in the PDF conversion. So instead of getting thousands of interactions, you now only get dozens of cell-cell interactions between the tumor and liver cells. The liver is consisting of a lot of hepatocytes, and we know that liver metastases are transcriptionally very different than primary tumors and, for example, lung metastases. So we wanted to study interactions between hepatocytes and tumor cells. So this is great. We have now a prioritized list of interactions, but it's still a very descriptive endeavor, right? We had a long list, and now we have a short list. So now we need to perturb to test if these are important and causing phenotypes. So how do we do that? Of course, first we have to switch to the mouse. So now we establish first a syngeneic colon cancer liver metastasis model. For that, we take intestinal cancer organoids, and here we can activate APC, P53, inactivate APC and activate KRAS, inactivate P53 and lose SMAD4. So we have an AKPS organoid, which are also important mutations in human cancer patients. If we inject this into the spleen, we get these macroscopically visible nodules within 2 weeks. So this is basically a highway where we have the portal vein, which will bring these cells into the liver, and we have these nodes. And also, if we section them and look at their microscopy, then we see that these look like adenocarcinoma that faithfully reflect the morphology that we want to see in colon cancer. It's still a model that we can control in vitro, an organoid, so we can add different tools. And the tool that we're using here is proximity labeling of the tumor microenvironment. So we use a slip cherry module, which is basically a secreted fluorophore, which then penetrates the neighboring cells in around 100 micrometers around the lesion. So here you see the lesion, and here you see the secreted cherry, which is highly expressed in the metastasis. But in neighboring hepatocytes, you see these little punctae, which means this is the absorbed cherry, which can now be used to flow cytometrically identify which hepatocytes were next to the metastasis. So that's our tumor model. Now we need to perturb. How do we perturb? Well, of course, we've heard today from Brian that there's a big problem if we take a cell line, perturb it, and put it into an animal, because we have this huge bottleneck effect. And we want to address many different perturbations in mouse, so that's a big problem for us. Additionally, if you put the guide RNAs into the cancer cell, you will get cell autonomous effects, mostly. And it's going to be hard to assess what we are interested in, namely cell interactions. So we developed an outside-in CRISPR screen, where we're actually perturbing not the cancer cells, but the host organ. And we call this a mosaic liver. So the idea is as follows. We have a mosaic liver with different perturbations at different locations. And if a single metastatic cell is now coming in, it might randomly end up next to a perturbation which is good for its outgrowth, and will then lead to a full-blown micrometastasis. If the cancer cell ends up next to a perturbation which is incompatible with its outgrowth, it's going to be depleted. So that's the screening model. How do we do this? This is all work by Costanza, an excellent former PhD student at the lab. She's now doing a postdoc in Rockefeller. So she basically takes guide RNAs in the format of transposon vectors and co-injects them with a Sleeping Beauty transposase. And the liver is a beautiful organ for that purpose, because you can, for example, use hydrodynamic tailwind injection and efficiently get genetic material into hepatocytes. So this Sleeping Beauty will stably integrate these guide RNAs into the hepatocyte genome. And we do this in the context of a CRISPR-A mouse. So we have a catalytically dead Cas9, which is fused to a transcriptional activation domain. And this whole thing is under the control of albumin-Cre. So albumin-Cre, and here we have a lock-stop-lock, so this is only activated in hepatocytes. And then, of course, we do this not with any gene, but we do it exactly with these genes that I showed you before, these receptor-ligand interactions that we found in human patients to be expressed at the interface between liver and metastasis. So how does this look like for one gene? Here I show you glutamine synthetase. Glutamine synthetase is usually expressed in one layer of hepatocytes only around the central vein, which you see here. So spatially extremely confined to one layer of cells. And now we bring in a guide RNA against glutamine synthetase, and we wait for 6 days in these CRISPR-A mice, and you see tons of ectopic expression all over the place. So this is one gene. Now we can do this for 1,000 guides in one mouse while still getting 1,000-fold coverage. Because the mouse has 100 million hepatocytes, and even if we do the screen currently at a 1 to 2% targeting, we still can recover 1 million of targeted hepatocytes, meaning we can see a guide RNA 1,000 times in one individual liver of this mouse. So this is the perturbation. Now we bring the two parts together. So we have the cancer model, these organoids, which label their microenvironment, and then we can use flow to sort the tumor cells because they're green and red, the nearby hepatocytes because they're red, and the faraway hepatocytes because they have no cherry. And we do this in the context of a mosaic liver, and then we sort the two hepatocytes, so the hepatocytes that were next and the hepatocytes that were far away from our metastases. And then we can simply PCR amplify the guide RNA, use next-generation sequencing, and quantify. So a lot of testing that this really works, which I'm going to skip. I'm just going to jump to the results, and this you see here. So this is now a plot of all of our perturbations ordered by enrichment. Here you have the ones that are mostly found next to metastases, and here you have the ones that are mostly depleted next to the metastases. And when we look at the gene names, we were excited to see factors such as EGF that we found are promoting nearby metastases. And down here you see, acute phase proteins such as SAA1 and so on. So that made a lot of sense in terms of directionality. But we were also very excited to see several molecules that are involved in axon guidance signaling pathways, such as plexin B2 and semaphorin, to be popping up in the screen. So initially we wanted to do some in vitro validation, which was actually much more challenging than envisioned, because cell-cell interaction modeling in vitro is usually quite artificial, or it's very complicated to manage for these in vitro validations. So we did some experiments where we used co-cultures of hepatocytes and hepatocyte cell lines, and basically did this overexpression of our targets, and then seeded the organoids on top of them, so that we can then basically quantify colony formation. And when we do colony formation, we saw that these plexin molecules, overexpression of plexin B2 in hepatocytes, increases colony formation of the organoids. This was a lot of work and hard to do, but it was also nice because it told us that this is actually an effect between these two components, because we only have hepatocytes and cancer cells. It means that there's no, let's say, immune cell or third cell type involved. It has to be a direct cell-cell interaction because we only have these two components in vitro. So this motivated us to pick this plexin B2 and move on to in vivo validations. So this is now work of Morgan in the lab. He now switched from hydrodynamic tail vein delivery to AAV, because now we want to perturb the whole liver. So now we use an AAV guide RNA, and we basically have the same albumin-free CRISPR-A mouse. We bring in our AAV, and then we wait for a week, and the result you see here, so you have basically 90% of the liver transduced, and when you see the control plexin at the cell surface receptor, here in white, and you see down here, this is the AAV targeted mouse, so you have a huge overexpression of plexin B2 in the whole liver. And at that stage now we come in with our intrasplenic injection of the cancer organoids, and then you see the results. So this is control mice, so the liver is not perturbed. You see a few macroscopically visible mets, and if we have now a plexin overexpression in the liver, you see a huge increase in metastatic load in these mice. Same cancer organoids, we only perturb plexin expression in the liver. And you can quantify this. Here you see it for these syngeneic colon cancer organoids. We also used KPC cells. We have also used a few other syngeneic models now, and we consistently see this increase in metastasis engraftment. What's different in these tumors? So we took some sections. Here you see these lesions in the plexin B2 overexpressing liver. And when we look at them, the size is rather comparable. So there doesn't seem to be a huge phenotype in terms of proliferation. but rather the frequency. So probably just the outgrowth efficiency is much better. And if we looked at the histology, we saw that the tumors that developed in these plexin B2 overexpressing livers appeared much more epithelial than the ones in the control livers. This prompted us to do single nuclei extractions and then do multi-omics, so single-cell RNA-seq and single-cell attack-seq profiling. And this gave us a big difference in KLF4 accessibility between in these tumor cells that we found in the plexin B2 overexpressing livers. And we also stained for a lot of markers such as EpCAM and KLF4 here. And we basically narrowed it down to this phenotype that KLF4 seems to, plexin B2 signaling seems to upregulate KLF4, which then seems to instill this epithelial fate in these tumors, which makes them more likely to be able to proliferate in the liver. So, of course, we don't want to increase tumor loads in human patients. So the next question was, can we do the opposite? And can we remove plexin and still get an opposite phenotype of what we have seen when we overexpress plexin? So for that reason, now we switch to the traditional Cas9 knockout mouse, and we create a plexin B2 knockout, also with AAV delivery of the guide RNA. The results you see here, so this is non-targeting control, a lot of plexin at the membrane, results with a lot of metastases. If we remove the plexin at the membrane, we abrogate the ability of these cells to metastasize. And this is not only the case in this intrasplenic model, we also now established an orthotopic injection model with colonoscopy. So this is rather laborious experiments where we have 3 operators that inject these cancer cells into the colon wall with a colonoscope. And then we can do this, of course, in the context of a mouse that is knocked out in the liver. So again, tumor cells are not perturbed. We knock out plexin B2 in the liver. You see the primaries. This is an early picture where we had an even bigger primary with plexin knockout, but we're not interested mostly in the primary. Now when we do many, many samples, we know that the size here is not different, but the big phenotype is this creates a lot of liver mets, whereas the knockout in the liver of plexin B2 also in orthotopic context prevents liver metastasis. So, last slide in terms of input. Of course, we then asked what's the interaction partner. Thanks to the database, we know it could be a semaphorin, and we saw that these semaphorins are actually expressed in our cancer model. And so we then created quadruple. The problem is the organoids have not only one semaphorin that could interact, but 4. So we created initially quadruple knockouts, quadruple knockdowns, and later on quadruple knockouts with Cas12, and did these competitive injection assays, and also here saw a phenotype that was mimicking the modification of the receptor on the side of the liver. And last but not least, if we look in human patients, we see that for 3 of these semaphorins, if their expression is higher in the primary tumor, this correlates with worse survival or worse time to metastasis. And also in these same patients, when you look at the liver mets, you see an amplification of copy number in these 3. And one of the 4, we don't see this association with survival, and we also don't see a copy number gain. So let me summarize. We have shown that we can use spatial transcriptomics to prioritize which ligand receptor interactions might be relevant and relevant for further experimental validation. We have established an in vivo cell interaction screen, and this screen can easily be adapted to other cell interactions by using other Cree drivers. And in terms of biology, we have uncovered that this plexin in hepatocytes seems to be a gatekeeper of liver metastases, and we hope that in the future, by blocking this interaction, we might shield the liver from incoming mets. The paper has been published a few weeks ago, and please have a look if you're interested. So this is the general vision of the lab, is that we start from human patient material, do these cell interaction predictions, and then use our in vivo CRISPR screens to identify targets and hope that at some point we can cycle back to the patient. So with that, I'd like to thank all the people that were doing the work, the collaborators, the funding sources, the organizers, and all of you for your attention. I'm happy to take questions.